Welcome to a special police briefing at the Chromosome 12 Police Department. It is our duty to protect and serve. We here at CTPD are devoted to keeping the streets of the scenic city, consisting of about 132 million DNA base pairs, safe. Hopefully the Police Academy taught you a lot about what I'll be talking about today during this briefing, so you shouldn't have too much trouble keeping up. Today, we'll be looking at some nice locales, their locations, their normal functions, the mutations and the criminals that they create, and finally some of the poor victims of any of these crimes. Without further ado, let's begin. Case 1. The first gene we'll be looking at is called RPS26. It's located in downtown, right here, about here, at 12q13, but it's still a nice place nonetheless. Its formal name is ribosomal protein S26. Now let's look at it and take a look at its normal functions. When this place is crime-free, it usually provides instructions for making one of about 80 different ribosomal proteins. You remember what a ribosome is from crop training, right? Correct! The ribosome is also made up of two subunits, large and small. The protein synthesized by the ribosomal protein S26 gene is found in the small subunit. The ribosomal proteins are involved in ribosome synthesis. Many others signal in chemical pathways regulating cell division and controlling apoptosis. Do you guys know what apoptosis is? Correct. Now, let's get to the good stuff. The criminal that caused this mutation goes by the name of Diamond-Blackfan anemia. Quite a mouthful, huh? I'll give you a rundown of the sicko's modus operandi. At least 9 RPS26 gene mutations have been identified in individuals with Diamond-Blackfan anemia. Mutations in the RPS26 gene cause this, obviously. Mutations basically affect the stability or function of the RPS26 protein. Diamond-Blackfan anemia is pretty uncommon. It affects about 5 to 7 live-born infants per million worldwide. Basically, there's a shortage of functioning ribosomal proteins, and this causes the apoptotic rate of the cells to increase, causing a decreased blood cell count, as you can see here. Unusual cell regulation in conjunction with this causes health problems and unusual physical features. This mutation is congenital and caused by random mutations or errors in cell division. That was just one gene, folks, and we have thousands more located in just this one chromosome. As you can see, cops here are pretty busy. Now let's move on to the second case. This nice suburban community is called KCNA1. It's more formally known as potassium voltage-gated channel shaker-related subfamily member 1. It's located at 12p13.32 on the upper north side of town, right about here. Let's look at the normal functions. Basically, it's a pretty place, and when encoded correctly, it has quite a specialized function in neural cells in the brain. The citizens of this gated community are among the smartest people in chromosome 12. Let's talk about the functions in the brain. Firstly, it provides instructions for making potassium channels. These channels transport potassium cations into and out of the cells. They are key in the cell's ability to generate and transmit electrical signals. Specifically, this gene codes for making potassium channels found in neurons. Can anyone remember what these neural potassium channels do? Correct! The flow of potassium ions causes changes in polarity of the neural membrane, causing action potential in the axon. Now let's look at the criminal. The criminal we can usually see associated with this gene is called episodic ataxia, and it's usually accompanied by its partner in crime called myokemia. At least 20 mutations in the KCNA1 gene have been identified in people with episodic ataxia type 1. This is an autosomal dominant disorder. Some mutations in the KCNA1 gene prevent the assembly of functioning channels, while others mutate the structure of the channel. Specifically, the potassium channels are not present or greatly diminish in count, and the potassium ion flow is reduced. It's quite uncommon. It affects less than 1 in 100,000 people. This decrease in potassium channels subsequently leads to an overexcitement of brain neurons and causes changes in signaling. This causes brief, recurrent episodes of poor coordination and balance. And this can, as I said before, this can be accompanied by myokemia, which causes muscles to randomly cramp, stiff, and twitch. Pretty unpleasant, right? Alright folks, we've looked at two nice autosomal communities and how a crime spree can ruin them. For the last case, I'll be doing something special. Recently, we've joined forces with the XCPD, the X Chromosome Police Department, in order to take down some thugs up there. So today, we'll be looking at a case from their chromosome, the X chromosome. The gene we're looking at now is the arrestilus homeobox gene, also called ARX. I chose this place because it's really nice. I have some folks who live up there, and my family goes there some summers for vacation. It's located on the northern side of X chromosome city at XP21.3. It's a pretty far drive from here. Now let's look at the normal functions of the ARX gene. The ARX gene creates a protein that regulates gene activities. It is a Hox gene. Quick, kids, can you tell me what a Hox gene is? Correct. Specifically, this Hox gene is involved in the development of the pancreas, the gastrointestinal tract, the testes, and the brain. Now let's move on to the mutations. When a mutation at this gene occurs, the criminal that usually shows up is known by the name lissencephaly. He is also known to strike other genes when there are mutations there, so this guy is one tough cookie. Basically, more than 20 mutations in the ARX gene cause lissencephaly. The mutation results in a non-functional ARX protein. It is an X-linked condition. The ARX protein can't perform its role as a Hox gene. The exact number of victims is not published, but is one of the rare sex-linked conditions. Since the ARX protein can't perform its role as a Hox gene, this impairs normal brain development, disrupts normal function in the pancreas and testes. And the signs and symptoms of this genetic disease are ambiguous genitalia. Alright, we've covered 3 cases, so that concludes the briefing. As you can see, genes are amazing and integral for survival, when they're functioning normally. But when a criminal shows up, a lot of things can go awry. Sadly, there are millions of mutations that occur every day. Luckily, our police departments and other chromosomal police departments work hard to protect you from most. However, sometimes they slip through. It is important to stay confident. Always remember our motto, you are here to protect and serve. Understand? Good. You are dismissed.